Lesson 3C, Gene Expression, also known as protein synthesis. Transcription. We know that DNA is the genetic material of the cell. Among other things, it stores all of the instructions for proteins. We also know how fragile DNA is, and for that reason, DNA is exclusively found within the nucleus. However, our familiarity with the cell's organelles indicates that protein synthesis occurs in the cytoplasm. To help solve this issue of protecting the DNA in the nucleus, while being able to have DNA instructions be transmitted to the cytoplasm, a copy of the DNA is made in a process called transcription. Transcription is the copying of the DNA segment that contains the instructions for the synthesis of proteins. Humans have thousands of these DNA segments encoding for proteins. We call these genes, while other segments of DNA do not seem to hold any instructions at all and sometimes are called junk DNA. This is a picture of human chromosome number 2. It has 243 million base pairs, and along it there is close to 100 genes encoding for proteins. In order for protein to be made, the DNA instructions must be copied or transcribed. This transcribed copy is made of RNA and is called messenger RNA, or mRNA for short. The mRNA will then travel out of the nucleus into the cytoplasm to be read by ribosomes so that proteins can be generated in a process called translation. In this lesson, we'll focus on transcription and we'll save translation for our next lesson. Transcription occurs in 3 steps. Step 1, initiation, where RNA polymerase binds to a promoter sequence on DNA, which essentially is the start sequence of a gene found on a chromosome. RNA polymerase will then unzip the DNA by melting the hydrogen bonds between the complementary base pairs. Step 2 is called elongation. This is the polymerization of the mRNA strand by RNA polymerase, making a complementary copy of the strand of DNA called the coding or template strand. The new mRNA strand will grow continuously from the 5' to 3' end, meaning that it will read the template strand in the 3' to 5' direction. As the RNA polymerase moves along the DNA, the complementary base pairs of DNA reform their hydrogen bonds, so there is no forking of the 2 DNA strands as we see in DNA replication. Rather, what we see is more of a bubble shape, hence the term a transcription bubble. The final step in transcription is termination. The RNA polymerase will reach a termination sequence on the DNA, and it will stop adding RNA nucleotides to the growing chain of mRNA, and it will simply fall off or dissociate. This image also shows the 3 steps in DNA transcription. 1, initiation, 2, elongation, and 3, termination, leading to a completed mRNA transcript. This image shows how the RNA transcript grows in the 5' to 3' end, reading the DNA template strand in the 3' to 5' direction. The new mRNA strand is called a primary mRNA, and it will undergo one last step before becoming mature RNA. In this step, an enzyme called ribozyme will cut out or cleave portions of the mRNA that are not needed. These sections of RNA edited out are called introns. The remaining pieces, called exons, are synthesized together and are allowed to exit the nucleus to be read and translated into proteins by ribosomes located in the cytoplasm. This brief video will summarize the transcription process.
The synthesis of messenger RNA is called transcription. Transcription begins when RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. A particular subunit of the messenger RNA, called the sigma factor, participates in recognition of the promoter region. Soon after transcription is initiated, the sigma factor dissociates from the RNA polymerase. RNA polymerase moves along the template strand of the DNA, synthesizing the complementary single-stranded messenger RNA molecule. Synthesis is in the 5' to 3' direction, with new nucleotides being added to the 3' end of the growing messenger RNA molecule. As the RNA polymerase advances along the DNA, it melts a new stretch of DNA and allows the previous stretch to close. When RNA polymerase reaches a specific sequence of nucleotides on the DNA called the transcription terminator, a hairpin loop structure forms in the messenger RNA, causing the RNA polymerase and the messenger RNA to dissociate from the DNA.